with what we're doing at Dec Bio is really trying to have this much more significant effect in terms of potency, expanding beyond that, 40% type of response rate, and then also going into major indications. And how we're doing that is we've taken a very different approach than everybody else that's out there. Is what we do is this approach called multi-peptide MHC targeting. Now's the time for solid tumor T-cell engagers. We've taken all these learnings and lessons, and then, with Dec, we're completely differentiated. Nobody else is doing this kind of multi-targeting approach. Key difference for what we're doing is that we're hitting these 4 distinct flags with one T-cell engager. And by hitting these 4 distinct targets that are all cancer specific, it allows us to have a lot more opportunities because you're covering a much broader patient population through this.
Jack, welcome to the show. How are you today?
Chris, thanks so much for having me. Doing great here in Kendall Square
In Kendall Square up in Boston.
in Boston. And it's been a great summer so far, but we're expecting a nice thunderstorm this afternoon. But yeah, life's good down here in the Biotech Center.
Well, time of this recording, we're just underway with the FIFA World Cup, and I know Boston's a venue. How's that impacting around town in Boston, Jack?
A lot of fun Scottish folks. I think all the bars dry, here in Boston, but it's been a lot of fun, and I haven't had a chance to go to any yet, but yeah, a lot of excitement around the town.
Well, yeah, and you're in Boston now, but I mean, you went to school at Johns Hopkins. You've been out on the West Coast. You've been on both coasts. You've been at where I know earned graduate degrees. So I thought maybe you could just talk a little bit about the time you spent at Johns Hopkins just to get us started. And I know you're working in a clinical kind of capacity, and sort of take us from there, and then what inspired you to leave the lab in Dec Bio a couple of years ago.
For sure. And so I did my master's at Johns Hopkins and knew I wanted, in the medical field. I wasn't really sure exactly if it was, MD, PhD, MD, PhD, all of those options. And then I went to, I wanted to work with patients, see what that experience was like. Knew I didn't want to be in the clinic every day, doing that as a time job. I knew I wanted to kind of be more in helping new ones. But ultimately, after my master's, I worked for a couple of years as a clinical research coordinator, and that was at Edison. And for those few years, I was enrolling patients, boots on, with consent forms, acting with the doctors every day, and very familiar with the clinical, frontings in oncology. And we were mainly focused at that time on metastatic prostate cancer. And, again, really wanting to take that experience. And ultimately, I came up with an idea for a drug. And so I specifically applied to PhD programs where I could work on this kind of idea that involved protein engineering. And I had no lab experience really, a minimal. And ultimately went and learned how to do, phage display, E-surface display at Stanford at PhD in immunology. And then with Dec Bio, as I was, wrapping up my studies there at Stanford, seeing what was happening in the clinical space, we just were having the first FDA approval for T-cell engager for solid tumors. And what a T-cell engager is, it's this simple kind of bispecific protein molecule where it looks like an antibody for most of these things. It's that classical Y shape. And what happens is one of the arms of the antibody, this bispecific antibody, is binding to one cell, in our case, a cancer cell. The other arm of the antibody is binding to a T cell, which is a type of immune cell, and ultimately forcing this artificial synapse that bridges them together and forces the immune cell to kill the cancer cell. This has been around for a long time with hematological malignancies, blood cancers. And then in 2022, we had the first ever approval for one of these types of molecules in solid tumors, where you're talking about, we have cancer. And so ultimately, really wanted to take clinical insights that I was having and apply some of these new protein engineering techniques and make this new class of medicines that I'm excited to talk more about later in the show here, but really expanding this usage of T-cell engagers to major solid tumor indications. And so we started in September of 2023 and based here in Boston, and yeah, I've been off to the races ever since.
So tell us a little bit about Dec Bio then. What is your mission and your platform? What is it you're trying to achieve?
One of the things that we're seeing with this initial approval for this first drug called Kimtrack, it has this ability to save lives, and they showed in a phase 3 setting that significantly is extending patients' lives compared to traditional chemotherapies, or even compared to these traditional immunotherapies. You know, despite that overall survival benefit, it didn't show that a response rate where when you're talking about a response rate in oncology, you're talking about tumors that are shrinking in a patient by greater than 30% is the definition of habits in oncology. And so in the phase 3 trial there, you were looking at about, high single digit, low double digit percentages. So very low, number of patients that have their tumors actually shrinking therapy. In 2024, there was an approval for a different medicine of this class, a drug called Imdeltra, or tarlatamab is scientific name. What they showed is a much higher response rate. 40% of patients responded to this type of medicine, and you also see that extension of patients' lives. And so we were looking to take from the learnings of those two medicines, where for both of them, they're applied to much smaller, patient populations. In the case of Kimtrack, treats about 1,000 patients per year between the U.S. and Europe. With this other medicine, it's, Imdeltra, also, sub 10,000 patients per year. With what we're doing at Dec Bio is really trying to have this much more significant effect in terms of potency, expanding beyond that, 40% type of response rate, and then also going into major indications. And how we're doing that is we've taken a very different approach than everybody else that's out there. Is what we do is this approach called multi-peptide MHC targeting. And so what we do is with our one T cell engager, we're able to recognize four different targets, four different flags that are on cancer cells. It allows us to have uh, greater recognition within a tumor where it's possible that might express one flag, other cells might express a different flag. And, with this multi-recognition event, that you're able to cover more tumors and then also more patients by, as a result of that. It's possible that you, Chris, could be expressing, target one, flag one, and I could be expressing target two, and both of us could respond to therapy. And so really having this a way of overcoming tumor heterogeneity, patient heterogeneity, really, having greater durability in terms of responses to these types of therapies. And we believe, and what's been shown is that for patients that have this greater than 30% tumor shrinkage, that does correlate with better extension of life, focused on having, uh, you know, focused on shrinking tumors and then also.
You're describing like in a way like truly a personalized medicine for each individual. I'm wondering before we go any further if you could just explain in plain terms. You already kind of gave a definition of what a T cell engager is, but for maybe those that aren't as who don't necessarily understand all the different cancer treatments, how is that different from other types of cancer treatments and targeted therapy now that patients might be familiar with?
For sure. So there's been the Nobel Prize for immunotherapy, where it was mainly focused on what's called checkpoint blockade, looking at these brakes on the immune system, and people have developed antibodies that are unleashing those brakes, and then the immune system to recognize cancer. That works in about 20 to 30% of patients. And the great thing about immunotherapy compared to something like chemotherapy is that with chemotherapy, you're only seeing these tumors shrink as long as you're being treated with chemotherapy. As soon as you stop the treatment, whether it's chemo, ultimately, once you stop treatment, the tumors will grow back out. With immunotherapy, the hope in it, and what we've seen in some patients is that you can have this tumor shrinkage. If your tumor can be completely shrunk, go completely away, 100% response, then it's possible to have this, immune effect where your immune system is now able to recognize that type of cancer, recognize those types of cancer cells, and you can have called the tail of the curve, where patients are in this 20 to 30% of patients having durable benefit beyond stopping treatment. And so that's really this holy grail of cancer medicine is you can treat to the point that a patient has a response, and then their immune system can keep the cancer in check and fit. On the flip side of this kind of off medicine are these T cell therapies where you're taking out cells out of their blood in a very complex processing where you have to do some preconditioning. You have to then, harvest the cells. You then have to do additional types of chemotherapies in between, and then you infuse these immune cells. But these are great, types of medicines for blood cancers. You see, very high response rates. 80 to 100% of patients are medicines. Again, mainly in the hematologic base. In solid tumors, we haven't really seen much these types of medicines. And so that's where T cells engagers have come into play, where it's kind of in between this checkpoint blockade and this where you only see this 20 to 30% of responding to checkpoint blockade by having this artificial recognition, engaging with your immune cells and engaging with cancer cells. You're able to create this artificial recognition of your immune cells and system against cancer. You're able to engage this remaining population. And that's focused is creating these molecules that can help patients that are not other types of medicines. And the other, part that people have been exploring is combining things, you know, putting multiple medicines together to then have even greater benefit and really, at the end of the day, really focused on extending patients' lives, giving them additional timely and I was on the front lines, Johns Hopkins, and so I've experienced, every patient that I enrolled in a clinical trial at that time has since passed away. And so, uh, me in terms of making new medicines, because I've seen families are going through, and I felt very hopeless when I was on that side of things because you can only prescribe or treat patients with what's available. And so that's really why I wanted to learn how to make new medicines and then now make medicines to give more things in the arsenal receive.
Clearly a huge unmet need in solid tumors, new immunotherapies and approaches to harnessing the immune system. But can you explain why it's been historically difficult for immunotherapies to get them to work against solid tumors? You mentioned that they've worked really well in lots of the blood types of cancers. So why has it been so difficult to work in solid tumors?
So when you are treating often with these blood cancers, you are hitting these targets that are also expressed on healthy blood cells. But it's possible that you can deplete all of your cancer cells and deplete all of those blood cells and still be okay. With solid tumors, the targets that you're going after, these flags that are expressed on the cells, for solid tumors, those are often also expressed on healthy tissues. And so if you think about the classic target that everybody thinks about in solid tumors is this target called HER2, very established in breast cancer. There's multiple major blockbuster drugs that have gone after it. People have tried engagers against it. And when you have this very potent immune recognition of something like HER2, very highly expressed in breast cancer, but then also has low levels of expression in other parts of the body, it has low levels. It's been shown that it has low levels of expression in the heart, such that if you have a very potent T cell engager, something that is one of the most potent medicines that we have in our arsenal, that you get these cardiotoxicities, you have your heart, and you can't ultimately dose through that in terms of having activity against the cancer, but not having off tumor activity against healthy tissues. And so in terms of what's been shown to be successful in this space. Again, there's been many failures. People have tried for a long time, decades to develop T cell engagers against solid tumors. Many failures there. What's been shown with these two successes is that the choice of the target that you're going after for these solid tumors, it's very important that that target has clear tumor specificity where it's only expressed on the cancer cells and not healthy tissues. If it is expressed in healthy tissues, it has to be a certain level. It can have a little bit of healthy tissue expression, but not very much because you have any type of toxicities. any of those toxicities ultimately prevent you from treating through and getting an effect in terms of a response, in a patient in terms of their cancer responding. And so what we've focused on at Dec Bio is really these cancer specific targets and, differentiating in terms of how we're going after those.
Let's turn to the pipeline now. Your lead asset, DBX01. How are you translating this biology and willingness to want to sort of target the immune system and capitalize on the power of T cells to tumors? You're looking to develop the asset in multiple different solid tumor types. Can you walk us through your thinking there and how you're looking to bring a product into the clinic?
For sure. And in terms of you know, what this initial approval of a T cell engager in cells was for. It was for a target that is expressed inside of the cell on the cell surface, the target that they were going after. How they were able to recognize that with a soluble T cell engager that's outside of the cell is that in case of cancer, there are certain proteins, certain targets that are expressed inside the cell that get presented on the cell surface in the form of this molecule called peptide MHC. This has evolved from bacterial, viral immunology. If a cell gets infected with something, then the immune system is able to recognize a flag that's on the cell surface from inside of the cell. And so what happens in cancer is there's the similar flag that gets presented on the cell surface from something being happening inside of the cell. When you look at the majority of cancer specific targets, they're expressed inside of the cell. And so what we do at Dec Bio, access these, intracellular proteins are what they're called, through these multiple flags that are being presented on the cell surface. The key difference for what we're doing is that we're hitting these four distinct flags with major. And by hitting these four distinct targets that are all cancer specific, it allows us to have a lot more opportunities because you're covering a much broader patient population through this. And so again, as I was saying earlier, it's possible you could express one of the targets, I could express another, and both of us could respond to therapy. Some of the cancers that we're focused on, one of the targets might be more preferentially expressed, while a different cancer, a different one of those targets might be more preferentially expressed. And so through this multi-targeting, we're able to cover more cancer types and then also more patients. And within those patients and those cancer types, more of the exact, cancer cells within their tumor. So really focused on having this effect where, through this multi-target recognition, we're able to get broader addressable patients in terms of, who could actually receive this type of medicine, and then also having this stacking effect in terms of having more potent and deep responses in patients that are all cancer specific because these targets are not expressed in any other health.
So how has that informed your prioritization to go into like lung cancer, let's say first in trials? I mean, there's theoretically a lot of different tumor types you could be
Mhm.
evaluating DBX01. So I know you're also looking at other GI types of cancers like gastric and esophageal. So tell me a little bit about how you're looking at prioritizing the different tumors. Is it driven by biology, immune phenotype of the underlying
Yeah.
tumor?
Spot on. So so we've built out this, prioritization analysis that we've built internally where it's looking at the target biology in terms of, how prevalent are the targets, how highly expressed they are above this response threshold that we've identified. Is that tumor type considered immune cold or immune hot? Are there immunes in that tumor? Such that we could engage, the cancer cells with this type of T cell engager modality. And then also looking at, the clinical path of development, how many other molecules are there in development for completely distinct, talking about uh, cancer. There's many types of small molecules, oral pills that people are taking, radiopharmaceuticals, antibody drug conjugates, CAR T, et cetera. And so kind of more what's going to give us that clear signal early on in the clinic that we can say this molecule is very valuable, let's say for indications. So really focused on having a very clear path in terms of our clinical development that we can have a very capital efficient plan. And then once we have that safety signal, that efficacy signal, then doing a more comprehensive development. That's kind of what we saw with anti-PD-1, in melanoma, and then went after lung cancer, and now, 18 plus cancers that these anti-PD-1, and so, um, you know, really focused on having that initial signal and then getting from there.
What gives you hope that your T form um, could work where previous attempts to develop assets in solid tumors beyond the couple of examples you gave of products that broke through? I mean, you mentioned the f targeting four different flags, but what gives you hope? Is it that what gives you hope, or are there other things behind the scenes, data that give you hope that this is going to be successful?
We have been building this nice preclinical package, comparing our molecule against other, molecules that are in the clinic and showing, a multiple logs order of improvement in terms of activity. So where other failed in a certain tumor type preclinically, we're showing activity. And so really focused on, using this multi-targeting, again, with the traditional approaches, if you're missing that one target that you're going after, then that patient's not going to respond. And the same goes in when you look at things preclinically. If a cancer cell that you're treating, a cancer type that you're treating doesn't express that target, you're not going to see a response. With our multi-targeting approach, some cancers might be expressing one of the flags more strongly versus others might be expressing the other one. And so by having this more multi-targeting coverage of more patients, more cancer samples, and then, we've also done some additional innovations in terms of the molecule side, in terms of what it looks like. In this class of recognizing peptide MHC as a target, you often have these kind of wonky formats, these wonky molecules going into the clinic. That puts a lot of pressure on the manufacturing side of things in terms of creating formulas that can be stable enough for your drug in terms of how it's dosed, all these complexities downstream that I was seeing when I was at Hopkins. And with Dec Bio, with my protein engineering background, with our team's, protein engineering background, we've really put the emphasis up front on having something that's very easy to manufacture, very easy to dose, creating additional conveniences for doctors, nurses, and um, and then also creating this potency part of the molecule in terms of having the optimal geometry, the optimal, how the molecule all comes together. Lastly, we've built out this internal platform. When we're talking about targets, there's a concern like, okay, you're hitting this one flag that's on cancer cells, there's a flag that's on a healthy cell. How do you make sure that you're not going to hit that? And then in the case of these peptide MHC targets, we're hitting these four different cancer targets. There's 14,000 healthy flags, somewhat similar. And so what we've built internally is this comprehensive screening platform that we call our screen, basically, where for every molecule that we develop internally, we ensure that we hit those four cancer specific targets, but that we also counter screen, ensure that we don't recognize those 14,000 healthy targets, and have this very strict specificity workflow to ensure, very high potency, but then safety bar. So we feel very confident at this point, I'll say, in the specificity for pieces, and then in terms of the format, in terms of its developability, really to get on the in the dune and see what we want to see.
Well, let's talk about that. I think from what I've read, you're looking to enter clinical trials sometime next year. So, what are the biggest milestones between now and then, and what's the critical jobs to be done before you can get into the clinic?
Sure. Great question. And so, still at this point, we still have a few molecules that we're evaluating, trying to narrow in on what's going to be the one that we take forward. And, once we select the one, then that's where costs really escalate for early stage biotechs. The next step, right now we've been producing these types of molecules all at a research scale. So we have, small scale molecules that we've been putting together. The next scale thing to do is produce it at a clinical scale, a clinical grade. And so for that, you need much higher, much larger, uh, systems with very high sterility. You don't want a bacterial or contaminating protein in this drug. So That's the key timeline for these types of medicines is once you say, okay, we've decided on our final molecule, then you kick off this manufacturing timeline that in our industry is about 9 to 15 months, depending on what contract manufacturer you go with. Nobody in our industry really does any type of in-house manufacturing anymore because it's so commoditized at this point. And so, we're looking at about a 12-month timeline once we select that final molecule. And then at the end of that 12 months, then that's when we would be starting to dose patients. And right now we're looking end of 2027 or early 2028 as when we would trial.
So why do you is the right moment for T-cell engagers and T-cell receptor-based therapies in solid tumors? Is it the convergence of technology, functional biology, insights, validation in the markets, giving, what do you think, why do you think this is the right moment, Jack?
Sure. Great question. You know, again, 2022, first ever T-cell engager approved. And, people didn't have the highest hopes for this because, again, it had very few patients that had that what's considered a response in the cancer field, but it did have this readout of benefit of overall survival. And then just 2 years ago, we had the second approval here. There's been a lot of learnings in the field. All of a sudden, after these two kind of approvals, you know, things are converting. You're seeing an inflection point. You're seeing more money coming into the ecosystem from investors, from pharma. There was just a $4 billion deal announced earlier this week. And so, um, now's the time for solid tumor T-cell engagement. We've taken all these learnings and lessons, and then, with Dec, we're completely differentiated. Nobody else is doing this kind of multi-targeting approach. And part of that, in terms of why the timing is right, is that there's also been on of this uh, specificity screening workflow. It was a lot of people had concerns, in terms of hitting one of those 14,000 healthy flags. So with our internal platform that we've developed here, using the latest protein engineering technologies, we're able to comprehensively screen and really ensure that safety angle while also having that multi-target angle. And then the key thing that enables us to have this multi-targeting piece is that there's been these advances in the past few years around AI-enabled structure prediction. And so what that means in our field is around when you look at these flags, when these targets, historically, you don't know what you're going against. There might be a structure out there of what the protein that target this flag that looks like. But now with AI, it's completely this new field of protein structure prediction that has the resolution that we need in order to see what this actually should look like in a patient. And so what enables us to do our work in terms of hitting these four distinct targets is that we had multiple levels of validation in terms of, at the protein level that these things are expressed, at the RNA level, in terms of the, um, peptide level that being presented. The key thing for us is also having this structure prediction part where these four targets that we're going after at the surface level, at the structural level, that they look similar enough and that we can engineer something to bind these four different flags that look similar enough that we can recognize them with one T-cell engager. And that's, all within the last few years. AlphaFold, when we were starting the company, it was very enabling for, being able to further validate that.
So if everything goes right for Dec Bio over the next couple of years, Jack, what does the future look like for the company, patients, and what do you most want listeners to walk away?
Great. Uh, as I was saying earlier, in terms of, what we're seeing out there now with, these next gen chemotherapies, next gen ADCs, radiopharm, et cetera, real on them, uh, with the future of what I hope for the future of cancer medicines is that you could receive these types of therapies up to a point and keep your cancer in check, and maybe frequently, or maybe you completely don't get any more type of treatment after you're in this kind of remission, long-term remission from your immune system, keeping things in check. I think we're still in the early innings in terms of able to do that. But I think these next generation T cell engager approaches are very promising. Really unlock those pieces, and I think you know one of the things in our industry is that with all these new drugs that are coming out and coming on the market, when you're initially testing these types of medicines, you're treating the very, very refractory patients, patients that have gone through 5+ lines of therapy. They might have had chemotherapy, they might have had a hormonal therapy, they might have had radiation therapy or radiopharmaceuticals. You're saying, okay, you've already blasted your immune system with all of these type of toxic agents, and then we're going to reawaken it with immune therapy, a T cell engager. I think where the future is going to be is really moving these agents into earlier lines of therapy where patients have that, healthier immune system. And, we're already seeing this there in hematological and blood cancers where earlier line, you're seeing very fit there. And I think solid tumors are ripe for, taking this similar type of. I think that combination therapies are also, earlier and then doing combinations earlier, where you're doing very aggressive treatment earlier. And one thing that's gotten a lot of excitement recently is this idea of using treatments before, like very, once the patient's diagnosed, there's this treatment window where you can, treat a patient for 4 to 6 weeks with one of these immunotherapies. Then you're able to generate that immune recognition event and then remove their tumor. And then what a lot of people are seeing with these therapies in about 20 to 30% of patients, when you go to resect the tumor, there's no tumor there. So it's called this major pathological response, a complete pathological response. And so I think that that's, a really exciting area in terms of doing combinations in that of therapy. Only certain tumor types you're seeing. In fact, one of our uh, key scientific advisors, Patrick Ford, he led the early trials for this type of therapy with anti. Um, we're going to see more of that in terms of early aggressive therapy to that cure, where you don't see patient downstream. And I think with immunotherapies, that's those earlier treatments, that's really where it fit in well, versus after chemotherapy, it's a little bit tougher in the immune response.
Well, that's a very optimistic look at the future and the power of harnessing the immune system. Jack, I think you mentioned earlier how you hinted to how you're using AI in some of your functional biology work. I think I'd like to kind of close out with one final question. I just added this to the list of questions that I like because AI is everywhere. So
Yep.
I'd love to know, as a CEO, there's so much information that you need to stay on top of across the evolution of healthcare and oncology trends and things. So, how are you using AI right now just to stay up with everything that's going on in the field, especially given your leadership role and your time?
Yep. I think that's where I is the strongest. I will caveat all of this with you do have to be careful with AI. It can spout information, give you the wrong resource. You look at it says, okay, this is the standard of care for, lung cancer. And then you look at the sources and it's looking at clinical trials in esophageal cancer. And you're like, trust this.
Good
Again,
point. Good point.
very much a caveat, but looking at ChatGPT, Claude, Gemini, we use all of these things on they can't not. It's become the backbone of a lot of biotech in terms of being able to stay on top of what's the latest. Another one that is very commonly used in the medical field is open evidence. I think that's really much stronger in terms of the citations because doctors are actually using this to make clinical decisions. And so using that as a resource to say, okay, what's the standard of care? What's, on the horizon in terms of next gen therapies? And then where could our therapy fit in to the treatment paradigm? I think open evidence is much better ones. And then you're able to actually have the clinical trials cited that um, again, structural prediction. I think AI is touching every aspect of the drug development workflow. You're seeing it at the early, ideation, target discovery side of things. You're seeing it at the binder, molecule discovery side of things, the molecule things. You're also seeing it shorten down CMC time potentially through optimizations there, and then also clinical trial design, and then even the commercial side of things. I think it's touching the whole spectrum in terms of staying on top of, what's the latest in the field. Again, I think, use it to say, okay, what's coming down the pipeline? What do you see as kind of the next gen? And I think the key is also not using one of these as your kind of be all end all. I think using each one will spit out a different answer, and being able to use, you know, each one as a reference. One thing that I love to do is copy, the output from Claude and paste it into chat. What do you think about this? And they'll always say, "Oh, I think chat's not as good as me," or so. And so it's always interesting. But it's just an additional sounding board. It used to be we'd have these biotechs that would have 50 to 100 employees. You'd have these scientific advisory boards. Today in 2026, you can get the same thing, same with the team of two or three. And so I think it's of uh, accelerating development of novel molecules. And so I think we're still at the early stages of it. I'm really excited for the future, and really excited for patients to be able to have, more options.
Yeah, I remember back in my career, they used to lock people away like on the regulatory and clinical teams when they're preparing like a BLA filing.
Yeah.
We actually at a company I used to work at, they literally had a suite where it was locked down and like for a month. You didn't come out till the BLA was ready. And like imagine the number of people hours going into preparing a BLA filing
Yep.
when I have to, like, I haven't seen data yet, but this to me is one of the ripest use cases for AI because
Yep.
you've got your template, you got your FDA template.
Exactly.
It's like, let's just,
We've got all of this historical data. Let's like ideally it could just be copy paste. I think there was actually a company that just came out of stealth this past week that's trying to do that for IND filings. I think we're early innings of this, but I would not be surprised in the next few years
yeah,
if, it's the standard.
should save a
And
lot of time. And
uh,
you're coming into the clinic at the right to be able to maybe, utilize some of these tools. But
we're
thank you.
there. Very,
Yeah,
very thankful for that. And the hope is, I think, yeah, it used to be months long review of these
right.
100-page documents.
Good.
Ideally, they can just, put it in their own LLM and say, okay, where are the flags? And they can focus their efforts a little bit more. Again, I think that there's movement in that direction.
I can't wait to see the data in a few years on
Yeah.
how like new submissions,
Me,
because
me too.
yeah, yeah, like I said, you're coming into this at the right time. I mean,
Yep.
you may benefit from this, your company, as these regulatory timelines shrink down. But really exciting stuff. I'm super passionate about the, immunotherapy space. Jack, it was a real pleasure meeting you and getting to learn more about Dec Bio.
Really appreciate you having me on the show, Chris. Thanks so much for making the time.
Take care. Enjoy the rest of your summer.
If you enjoyed this conversation, be sure to subscribe to the RX for Biotech podcast to hear directly from the leaders who are shaping the future of personalized medicines. And if you or someone you know is navigating a care journey, be sure to visit therapyforme.ai to search for a specialist near you. And if you need that extra support, be sure to schedule a virtual telehealth appointment with one of our nurse navigators who can help answer your questions. Thank you for listening.